So now let's start our discussion regarding positive regulation. Okay, so before going to discuss about the positive regulation, there is a thing that I want to clear, that lactose operon. Okay, by nature, what is the nature of lactose operon? Lactose operon simply tries to suppress itself with the help of regulatory protein, right? As we have seen in the negative regulation, that the regulatory protein synthesized by lac-I, which is in low concentration, will bind with the operator region of the structural gene. Okay, and this is a by default choice. Okay, because by default that repressor protein is active, and after synthesis it just go for the binding with the operator region of the structural gene. Okay, so now as it usually try to suppress itself, right? So what can we say about the lactose operon? Because whenever lactose will be present in the medium, it become induced and start the synthesis of, or start the lactose operon, right? So what we can say? We can say that lactose operon is an inducible operon. Okay, because it become induced when lactose is present in the environment. Now let's start the discussion about what is going on in case of positive regulation. So this is again the structure of the lac gene. So here this is the regulatory part and this is the structural part. Okay, regulatory part again lac operator and structural part poses operator. Regulatory part's promoter is weak. Okay, so the expression of lac-I will be in low concentration. So the first thing, RNA polymerase with the help of sigma factor find out the promoter in the regulatory part. Okay, so after that RNA polymerase start its transcription, and via that it will lead to the formation of active repressor initially. Okay, but whenever lactose is present in the medium, remember there are small amount of trans-acetylase present in the environment. Okay, before lactose operon starts. Okay, that means there are small amount of synthesis of beta-gal permease and trans-acetylase is going on, although the lac operon is off. Okay, so this small amount of trans-acetylase will convert the repressor protein into inactive repressor protein. Okay, remember one thing, only just lactose do not have the ability to repress the repressor protein, but lactose will first react with the trans-acetylase and converts itself into allolactose, and this allolactose will inhibit the repressor. Okay, so what is the utility of that? So as a result the repressor become inactive and they no longer unable to bind with the operator side. But there is a question, what about that repressor that are already in interaction with the operator? So here also the allolactose will inactivate the repressor, and once they become inactivated they do not have the affinity towards the operator, and as a result they will simply comes out from the operator. So now RNA polymerase have its way to start the transcription, and lac-Z will eventually synthesize beta-galactosidase, lac-Y will synthesize the permease, and lac-A will synthesize the trans-acetylase. So what is the main idea? Main idea is, although the lactose operon is not active, there is a very small or minute amount of synthesis of beta-gal permease and trans-acetylase is going on. Okay, constitutively. Okay, so whenever there is a presence of lactose in the environment, first of all it will convert some lactose that is available in the cell into allolactose. Okay, so that it can inactive the repressor. Okay, so after that it cannot bind with the operator of the structural gene. So RNA polymerase now have the ability to start the transcription, and those repressors that are already in binding with the operator also become inactive with the help of that allolactose, okay, and as a result they comes out from the operator, okay. So lactate gene will leads to the formation of beta gal, and permease will be synthesized by lac Y, and trans-acetylase will be synthesized by lac A. So up to what time the lactoperon will be one or will be on, and how it again become repressed or suppressed. So remember one thing, after a certain time when bacteria can interpret that there is no longer lactose left in the environment, the beta galactosidase that usually involved in conversion of lactose into glucose and galactose also start utilizing those allolactose, okay. So it, as allolactose become degraded, this repressor again become active repressor, and they will bind with the operator of the structural gene, okay. That is how the entire regulation mechanism is going on, okay, and that is how lactoperon regulates from positive to negative depending on the lactose is present in the environment or not. If lactose is present, positive regulation; if lactose is absent, negative regulation, okay. How? Whenever, let me repeat it, whenever lactose is absent in the environment, the repressor synthesized by lac I is the active repressor. It will bind with the operator position of the structural gene and prevent RNA polymerase from transcription, okay. But whenever lactose is present in the environment, the trans-acetylase will first convert some initial lactose into allolactose, okay. Now this allolactose will inactive the repressors, and there another important thing that you need to remember, okay. So this another important thing is some lac Z, lac Y and lac A, and there is a very small amount of synthesis of lac Z, lac Y, lac A constitutively, okay. So that is the reason why trans-acetylase have the ability to utilize the allolactose, to convert lactose into allolactose that can be used to repress the repressor, okay. So as the repressor become repressed, okay, so lactose operon become induced to start, and RNA polymerase will start the synthesis of beta galactosidase, permease and trans-acetylase respectively. So that it for today.